It started.
Okay, are we ready for that? Okay, I think I'll kick things off. Good morning, everyone, and welcome to the interdisciplinary session of our second annual Oxford-Cambridge Student Symposium, Varsity Sci. I'm Shreyas, President of the Cambridge University Biological Society, and it's a great privilege to host Professor Peter Hore from the University of Oxford today. Before I introduce Professor Hore, I have a few words from our sponsor, BitBio. Today, we have a unique opportunity to build a bright future. To do so, we need to extend our technical repertoire and transition biology to engineering. Tapping into the potential of biology will accelerate biomedical research, enable new generations of cures, and open up new avenues for increased global sustainability. Founded by Dr. Mark Cotter and Florian Schuster, BitBio is an award-winning human synthetic biology enterprise. BitBio's mission is to code cells for health. To do so, they apply the principles of computation to biology. Their current focus is to develop a scalable technology platform capable of producing consistent batches of every human cell. So now I'd like to introduce our speaker. Professor Peter Hore has spent most of his life in the Department of Chemistry at the University of Oxford, first as a student, then as a junior research fellow, and now as Professor of Chemistry and Fellow and Tutor of Corpus Christi College. His only time away from Oxford was a postdoctoral fellowship at the University of Groningen. Over the years, he has worked on a number of topics around magnetic resonance and the effects of electron and nuclear spins on chemical reactivity, an area known as spin chemistry. These include spin hyperpolarization, protein structure and folding, photosynthetic energy conversion, and NMR methodology. Since about 2005, he has been trying to unravel the biophysical mechanism that allows migratory songbirds to detect the direction of the Earth's magnetic field as an aid to navigation. And this is why we thought he would be our perfect interdisciplinary keynote speaker. So this talk, as usual, will be followed by a Q&A session. If you're on YouTube, feel free to post your questions throughout the talk. We will compile them and pose them to Professor Hore. If you're on Zoom, you can either ask the questions yourself using the raise hand function will allow you to speak. You can post them in the chat, or you can send them privately to me or any of the other moderators. So without further ado, I'd like to welcome Professor Peter Hore.
Great, thank you very much, Shreyas. I'm delighted to be able to talk to you today. Right, let's try and share the screen. Is that working?
Yep.
You can see my first slide, good.
Yes.
Great, so good morning. Right, I'd like to start by asking you, just for a few minutes, to imagine that you are a bar-tailed godwit, a medium-sized wading bird. You've spent the summer in Alaska, but it's starting to get chilly. You need to fly south for the winter, and your destination is New Zealand, 11,000 kilometers away across the Pacific Ocean. But that's not a problem. You can fly at an average speed of 60 kilometers an hour, day and night, non-stop. So all being well, it'll only take you 8 days. So that, if you like, is the easy bit. More challenging, perhaps, is to know how to navigate. Well, you can use the sun and the stars to get a sense of direction. If it's not your first time, then you can use landmarks, but of course, there are not too many of those over the ocean. You also have a keen sense of smell, and that will probably help you home in on your target when you get close to it. But what you really need is one of these, a magnetic compass. Unlike the sun and the stars, the Earth's magnetic field is always there. And if you have a compass, then you should be able to get a sense of direction that will help you get to New Zealand. And what I want to talk about is how that magnetic compass works. And it's not just the godwits. Billions of small songbirds fly thousands of kilometers every year. Here's one of them, the northern wheatear, weighs about 25 grams. And there are populations that spend the summer in Baffin Island that fly to West Africa and back every year. Another population that goes between Alaska and Kenya. Extraordinary distances. Fascinating to know how they do it. And I would like to know how they navigate. Even the European robin. Most of the robins, of course, we see in our gardens in the UK are there resident all year round, but there are plenty of European robins that do migrate from the northeast of Europe and Scandinavia to the coasts of the southern Mediterranean and around Turkey and Israel and so on. So they all, it seems, detect the Earth's magnetic field and use it to get a sense of direction. And my colleagues and I in Oxford and my collaborators elsewhere have been trying to work out how this magnetic compass works. And it's been clear since the 1960s that migratory songbirds really do have this ability. So first of all, how do we know that birds like the robin actually have a magnetic compass? Well, I won't describe to you the experiments that were done in the 1960s that first convinced people of this remarkable sense. Rather, I will talk about how we do it nowadays. So this is a building on the campus of the University of Oldenburg in northern Germany, and it belongs to my close collaborator, Henrik Mauritzen, who is a biologist who studies magnetoreception, the mechanisms of detection of the Earth's magnetic fields, in particular in birds. And this house is made entirely of non-magnetic materials. We got the funding from a joint research grant from the American agency DARPA. And inside it, there are 4 chambers, 3 of which look like this. So each chamber is very carefully electromagnetically shielded so that the Earth's magnetic field can get in, but electromagnetic noise cannot. And inside this shielded chamber, there is this cage of magnetic field coils. So 4 coils in each of the 3 dimensions, 2 meters on a side. And by carefully controlling the currents through those coils, we can produce very homogeneous magnetic fields at the center of the coil system, which can be controlled in their strength and direction very precisely. In the middle of this coil system, there's a table on which sits 9 so-called Emblem funnels. So funnel-shaped cages at dusk, which is when these night migrants take a compass bearing, calibrate their magnetic compasses when they're in the wild prior to flying through the night. Sorry, I lost my thread. We test the birds here, 1 per cage. There's a translucent lid on top of each cage to prevent the birds escaping, so light can get in, the birds can't see out. They have no directional information apart from the magnetic field that we subject them to from the coils. And this is a very convenient assay for studying magnetic orientation. The birds can't get directional information from anywhere else. And it's well established in many laboratories that during the migratory seasons in the spring and the autumn, the direction in which the birds jump inside these cages at dusk is the direction in which they would fly if they were released into the wild. So you put the bird, 1 in each of these funnels for an hour in a particular magnetic field condition. And the way that the bird's orientational preferences are determined is by lining those funnels with scratch-sensitive paper. It's very low tech. And at the end of the hour, you take the bird out and you look at the paper, and schematically, it has a distribution of scratches, which, if the bird is well oriented, will give you a clear idea of where it wants to fly, in this case, towards the northeast. And then what you would do would be to repeat that experiment the following night with the magnetic field rotated, say by 120 degrees, anti-clockwise. And then if the birds try to jump in a westerly direction, then you know that they must be using the magnetic field to orient themselves. And that's how it was originally demonstrated in the 1960s. So this is a robust assay. It's been done, as I say, many times in different laboratories with different species of migratory songbirds. So how does this magnetic compass work? That's the question we would like to ask. And despite 50 years of research, we still really don't know. But I want to tell you about what is currently the most promising hypothesis for which evidence is gradually accumulating. Okay, so first of all, how doesn't this compass work? Well, this is a microscope picture of a magnetotactic bacterium, an organism that contains magnetic iron oxide crystals, which you see here as these black blobs. So this is biogenic magnetite. It's not something the animal has ingested. It's not contamination. Each of these blobs is a single domain of Fe3O4 magnetite, and they're aligned within the animal's body such that their magnetic moments reinforce one another. So this is exactly like a compass needle. This bacterium tends to align its body in the Earth's magnetic field, quite passively. It's not by choice, it just behaves like a compass needle. Of course, the colors here are false colors. So why would a bacterium need to detect the Earth's magnetic field? Well, these creatures live in shallow pools of water, and they need to distinguish up from down, because if they get too close to the surface, the higher oxygen content will kill them. They're too small to use gravity, so they need to swim along the lines of the Earth's magnetic field. Well, now, birds don't have a magnetic compass that works on the same principles. It would be difficult to imagine enough iron inside a bird's body that it would align itself like a compass needle. But there are two crucial properties of the bird's magnetic compass that argue against magnetite and in favor of the mechanism I'm about to describe to you. So if you imagine a bird in the northern hemisphere in the spring, when it wants to fly north towards its breeding grounds, then it experiences a magnetic field which points down and to the north. So there's a horizontal component in red pointing north, a vertical component pointing down, and the black arrow is the net field. Now, if you test these birds in the Emblem funnels and invert the horizontal component of the field, then perhaps not surprisingly, they try to jump in the opposite direction to the south. And of course, our magnetized compass needles would do exactly the same. They'd point south. If instead you invert the vertical component, so it points up, the birds still go south. And it's only when you invert both horizontal and vertical components, so the net field, the black arrow, points in exactly the opposite direction, only then do the birds go in the right direction. So this is not like a compass needle. It's not a polarity compass. It's what's known as an axial or an inclination compass. Very different from how a magnetized magnetite particle would behave in a magnetic field. And it's also light dependent. If the birds are tested in blue or green light, then they can orient in the magnetic field perfectly well. If they're tested in yellow or red light or in total darkness, then they jump in random directions in the Emblem funnels. They can't use the Earth's magnetic field. The compass doesn't work. So it's a light-dependent axial compass. And it's difficult to see how that could be explained on the basis of magnetite crystals. So the mechanism I want to tell you about is chemical. And it's based on a type of chemical reaction intermediates known as radical pairs, which we think are formed in a protein called Cryptochrome, of which more in a minute. So the basic idea is the following. We have reactants, somewhere inside the bird's body, which absorb blue-green light and are converted into chemical intermediates, known as radical pairs, which can then do two different chemical reactions. Either they can return to the reactants, or they can go forward to form a chemical reaction product. So all we need for this to constitute a magnetic compass, at least in principle, is that the branching ratio, the probability of going backwards or forwards here, depends on the local magnetic field. So we need a chemically sensitive, a magnetically sensitive chemical reaction, and one that's sensitive not just to the presence and strength of the field, but also its direction. And then all the animal would have to do would be to interpret the change in the yield of this reaction product as it moved its body around in the Earth's magnetic field, and that could give it a magnetic sense of direction. Now, at first sight, this seems a ludicrous idea. If you do the usual back of the envelope calculation of the energy of interaction of the Earth's magnetic field with a molecule, even a molecule like a radical that has a larger magnetic moment than a closed shell molecule, you'll rapidly come to the conclusion that that interaction energy is about 1,000,000 times smaller than the thermal energy, which is Boltzmann's constant times temperature. And on that basis, you would say there's just no chance that there would be a significant effect on a chemical reaction of such a weak magnetic interaction. And up to a point, you would be right, but there is a mechanism that involves radical pairs as chemical intermediates that for sure can be sensitive to such weak magnetic interactions. So a simple analogy may help understand this. Think of a chemical reaction which converts reactants into products as being equivalent to tipping over a heavy stone block. Now, of course, that requires a certain amount of energy. You have to raise the center of gravity of the block before it will tip over. And so imagining that a magnetic field might affect a chemical reaction is like imagining that a fly bumping into the side of this block is going to affect the rate at which it tips over. Just simply won't happen. The energy is far, far too small. But if you look at the same process a different way, suppose you've already put the energy in to get the block poised on its edge like this. Clearly a highly non-equilibrium state. It's teetering here. It could easily fall forward or backwards. Now a fly falling on one side of this block could make a lot of difference to whether it actually ends up going towards the products or back to the reactants. So the message here is that if you have a highly non-equilibrium state, then it can be sensitive to very tiny amounts of energy. And that's the mechanism I want to describe to you. And it's a real mechanism. So we've known about the radical pair mechanism since the 1970s. And it was in the 1970s, when the mechanism was in its infancy, that it was first suggested that it might be the basis of the bird's magnetic compass. Now, the mechanism since then has been shown to be absolutely genuine. There have been hundreds of studies of radical pair reactions in the laboratory, which have been found to be sensitive to weak magnetic fields. So it's a real mechanism. And the big question is, could it operate inside a bird's body? And could it provide a magnetic compass sense? So those are the kinds of questions we're trying to answer. Okay, so now a little bit about what this mechanism actually is and what radical pairs are. So this is a simple schematic reaction scheme. So we have a pair of reactants, A and B, or maybe two parts of the same molecule. And these are closed shell molecules. So all the electrons are paired up, and that's supposed to be indicated by these little arrows. So each electron, as you know, has quantum mechanical property called spin angular momentum, to give it its full name, and associated with that angular momentum is a magnetic moment. So electrons are magnetic. But if all the electrons are paired up, then their magnetic moments cancel, and there's not much magnetism left. Now, one of the properties of organic free radicals is that their reactions conserve spin. So if we imagine shining light on this molecule with a wavelength that will be absorbed by one of A and B, and that will trigger an electron transfer, say from A to B, then you will form a pair of radicals. Now, B will have a negative charge and A will have a positive charge, and the dots indicate the unpaired electrons. So we will have formed a radical pair by this spin-conserving, light-induced electron transfer. And this radical pair initially will be in a singlet state because spin angular momentum needs to be conserved. So the two electrons, one in each radical, separated by maybe 15 or 20 angstroms, will be antiparallel. But the radical pair doesn't stay in the singlet state for very long because there are internal magnetic interactions between the magnetic moments of the electron spins and the magnetic moments of certain atomic nuclei like hydrogen and nitrogen. And those so-called hyperfine interactions cause the singlet radical pair to convert coherently into a triplet and back again. So by triplet, we mean the two electrons, one in each radical, have parallel spins rather than antiparallel. And this is a quantum mechanical process indicated by these curly arrows. So it's oscillatory. The radical pair oscillates between its singlet and triplet states with a frequency of a few megahertz. So a time scale of hundreds of nanoseconds to microseconds. And that's because this singlet state is a coherent superposition of the allowed spin states of the radical pair. And that process is sensitive to external magnetic fields. So in just the same way, if you wave a magnet near a compass needle, then the compass needle will change direction and you could get it to flip over if you apply a magnetic field in the right direction. And that's what we want to do here. We want to influence this spin flip from singlet to triplet and back again. And that can be done by external magnetic fields, for example, that of the Earth. Okay, so what? We can affect the frequency and the amplitude of these oscillations. Well, if you now think about the subsequent chemical reactions these radicals could undergo, let's suppose the singlet radical pair could come back to the reactants, and the triplet radical pair could go on to form products. So both processes conserving electron spin. So the products would need to be in a triplet state too. So we have two competing reactions. And if by applying the magnetic field, for example, we increase the probability of the radical pair being in a triplet state, then more of the radical pairs will react to form the products, and fewer of them will be available to go back to the reactants. So by the time all of the radical pairs have reacted one way or the other, the final yield of this product C will contain information about the magnetic field experienced by the radicals. And then all you need is some way of interpreting that changed reaction yield, and you have yourself a compass. So that's the basic idea. And as I say, it's a real mechanism. It really does happen. And if the radicals are not too close together, so their interaction one with the other is fairly weak, then you can affect their interconversion with very weak magnetic fields, such as that of the Earth. Okay, so that's the principle. So when we started this, 10 or 15 years ago, we wanted a molecule that we could use to demonstrate this principle, that you could make a chemical compass. Because although there have been lots of investigations of radical pair reactions, none of them had investigated this possible directional dependence, and all of them had studied much stronger magnetic fields. So typically, first of all, the Earth's magnetic field is about 50 microtesla on average, varies a bit around the surface of the Earth. It's not very strong, but typically all the laboratory studies of radical pair reactions had started at several millitesla and had then worked up in magnetic field strength, often to several Tesla, the kind of magnetic fields used in MRI these days. We wanted to start at around 1 millitesla and work down to 50 microtesla. So we needed a molecule as a proof of principle experiment of making a chemical compass. And the one we chose is shown here. So it's a fairly unlikely looking molecule, but it proved to be a very good choice. Now, this shows you the structure a bit more clearly. What it is a carotenoid here, covalently linked to a porphyrin, covalently linked to a fullerene. So clearly not something you'd expect to find inside a bird's body, or indeed anywhere in biology. Although, of course, porphyrins and carotenoids are widespread in the living world, but not linked together like this, and certainly not with a fullerene on the end. We chose this molecule because we thought it would have the right photochemistry. And so it proved. If you shine light of the right wavelength on this molecule, the porphyrin will absorb it, and that will trigger 2 sequential electron transfers within the molecule. The first from the porphyrin onto the fullerene, and the second, so that produces a radical pair, and then the second electron transfer automatically follows immediately afterwards from the carotenoid onto the porphyrin radical. And that produces a radical pair, or strictly a biradical, because the reaction is intramolecular, with one radical centered on the carotenoid and the other about 4 nanometers, 40 angstroms away on the fullerene. And it's that radical pair that we thought would be sensitive to weak magnetic fields. And so it proved. So using this molecule and by making spectroscopic measurements of its photochemistry, measuring the yields of reaction products formed from this radical pair, we were able to show that a magnetic field of 50 microtesla, so comparable to that of the Earth, was able to affect the reactions of this molecule. And also, by aligning the molecule and then varying the direction of a weak magnetic field, we were able to show that it could act as a compass sensor. That is, the yields of the reaction products depended not just on the presence and strength of the field, but also on its direction. And that, of course, is what you want for a chemical compass. So it was light dependent, and we were able to verify, not that it was a great surprise, verify the theoretical prediction that radical pair reactions are not sensitive to exact inversion of the magnetic field direction. So like the bird's compass, rather than having a north and south pole, it's a bit like having 2 north poles. Exact inversion of the magnetic field had no effect on the magnetic sensitivity of this molecule, just like the bird's axial compass. Okay, so now this protein cryptochrome, this was first suggested in the year 2000 as a possible host for light-induced radical pairs inside the bodies of migratory birds. Cryptochromes were only discovered in Arabidopsis in the 1990s, and by the end of that decade, it was reasonably clear that they had the sort of photochemistry that might be suitable for this purpose of magnetoreception. That is, unlike every other known vertebrate photoreceptor, they form radical pairs when they absorb light. So cryptochromes were, as I say, discovered in plants, subsequently found in many, many other species, organisms, including humans, and in birds. Now, we know that the magnetoreceptors, whatever they are, in migratory songbirds, are contained in their retinas, in their eyes, probably in photoreceptor cells. Cryptochromes, there are 6 known cryptochromes in birds, and some of them, at least, do occur in photoreceptor cells in avian retinas. So, and although this was suggested 20 years ago, it's still the only game in town. There's been no other proposed radical pair magnetoreceptor in birds. It's still the only known vertebrate photoreceptor that forms radical pairs photochemically. That doesn't mean it's the right molecule, but it's the front runner. It's the only one we have to work with at the moment. And evidence is accumulating that it might be the right molecule. And I want to tell you a little bit about that. So if you, actually just before that, if you look very carefully in the middle of this representation of the structure of the molecule, its molecular mass is about 60 kilodaltons. You can just about see in the middle a turquoise bit, which is the chromophore, and then some yellow bits down here, which are involved in electron transfer. So all of that is shown much more clearly here in this partial structure. So in the middle of the protein, non-covalently bound in cryptochrome 4, one of the 6 cryptochromes in birds, there is a chromophore called flavin adenine dinucleotide, closely related to riboflavin, which is vitamin B2. It's a yellow compound, and it absorbs blue light, roughly the same wavelengths that the birds require for their compass to work. And leading out from the flavin in the middle of the protein to the surface of the protein on the right-hand side here, there is a chain of 4 tryptophan amino acid side chains. And what we've been able to show is that when you excite the flavin with a flash of laser light in the blue, this triggers 4 sequential electron transfers within the protein. So the first from the closest tryptophan, A, onto the flavin, that forms a radical pair separated by about 8 angstroms, center to center. That's immediately followed by a second electron transfer from B onto A, a third from C to B, and a fourth from D to C. So well within 1 nanosecond of the flavin absorbing a blue photon, we have a radical pair with one radical centered on the flavin and the other on the terminal tryptophan, about 21 angstroms away. So the 2 radicals are well separated. And we look at cryptochrome 4 because it's probably the only avian cryptochrome which binds this flavin chromophore. So the others have very similar amino acid sequences, but with crucial differences in the region of the protein that would bind this flavin. And it seems that only cryptochrome 4 has the chromophore. And of course, if there's no chromophore, then the protein is transparent, it doesn't absorb visible light, and there can be no photochemistry, no radical pairs, no magnetic field effects. Now, the cryptochrome from plants, from Arabidopsis, has only 3 tyrosines. And we know that its photochemistry is similar, 3 sequential electron transfers, and not 4. It doesn't have this tryptophan D. So when the sequence and the crystal structure of pigeon cry 4 was published a few years ago, we all got quite excited about this 4th tryptophan. Plants, of course, probably have no need for a magnetic compass. They don't migrate very far. Songbirds do. So maybe this was crucial to the operation of this protein as a compass sensor. Well, we don't know the answer to that question yet, but it looks like it might be important. So what sorts of experiments were we able to do with the purified European robin Cryptochrome 4 that my collaborator, Henrik Mauritsson in Oldenburg, was able to express in bacterial cell culture in high yield and with high purity? So in Oxford, using a variety of different spectroscopic techniques devised explicitly to study these photosensitive proteins and their magnetic sensitivity, we were able to show, first of all, that all 4 tryptophans are involved in electron transfer. So to do that, we did 2 sorts of experiments, one of which is an electron spin resonance experiment or electron paramagnetic resonance. So a form of magnetic resonance like NMR, but done with electron spins rather than nuclear spins, in which we can measure the distance between radicals in a radical pair by measuring their magnetic dipolar coupling. Just the strength of the magnetic interaction of the electron spin on one radical and the electron spin on the other. And that interaction depends on the inverse cube of the distance between the radicals. And so we can work out which of the tryptophans is involved from the distance, knowing the crystal structure of a bird protein. And what we found was the distance was around 21 angstroms, confirming that the electron does come from as far as the terminal tryptophan D. What we could also do was to study mutants of this protein in which, one at a time, each of these tryptophans was replaced by phenylalanine. Phenylalanine is another aromatic amino acid, but it has a different redox potential and it doesn't do electron transfer. So you can block the electron transfer at position A, B, C, or D. And what we found by studying those proteins was that the radical pairs that are formed when the flavin absorbs blue light were very short-lived. If, for example, you put the phenylalanine at position B, then the radical pair is the flavin with trip A. And that has an extremely short lifetime, far too short to be sensitive to the Earth's magnetic field. Similarly, if you put the phenylalanine at position C, then the flavin trip B radical pair is a bit longer lived, but still not long enough. And so it's only when you put the phenylalanine in the terminal position, so now you have a radical pair separated by nearly 18 angstroms, or you look at the wild type, so then it's the 3rd or 4th tryptophan paired with the flavin, only then do you get a radical pair with a long enough lifetime to be sensitive to a 50 microtesla field. And so it turned out when we did the experiments, we have spectrometers, different sorts, different spectral resolution, different time resolution, that can measure effects of weak magnetic fields on the subsequent reactions of these radicals. And we found that both radical pairs involving trip C and trip D paired with the flavin, these both are magnetically sensitive. They both show magnetic field effects, and we conclude that this protein could be suitable as a magnetic compass sensor inside the bird's eyes. Now, we actually think that both of those radical pairs are involved, and that, in fact, this 4th electron transfer is reversible. The electron is hopping backwards and forwards between these two. and spending most of its time on D, but about 10% of the time on C. And we think the advantage of that is that C, being closer to the flavin, is more magnetically sensitive, and D, being close to the surface, might be better for signaling. Now, of course, the protein has to do both functions. Signaling is important. So what we think happens here is something similar to the plants, which is having formed the radical pair, the tryptophan radicals then get reduced, probably by some external electron donor out here in solution in the cell. And then you're left with the protein with only the flavin radical. And this is a long-lived state of the protein, which, if it's like the Arabidopsis cryptochrome, will have a different conformation in the C-terminal domain. That will then change the ability of this cryptochrome to bind to other proteins. And that will be the start of a signal transduction cascade, about which we know rather little at the moment. So we think TRPD could be involved in the signaling because it's closer to the solvent. And we think this tyrosine here, which is only 3 angstroms away, could also be involved in the electron transfer. There might be a 5th electron jumping here, and then this is right on the surface, and so could more easily be reduced by external electron donors to give a signaling state with a different conformation and different binding properties to partner proteins. So that's how we think it might work. And then finally, we were able to do exactly the same sort of experiments, but now with the highly similar cryptochrome 4 from 2 non-migratory birds, chicken and pigeon. They share exactly the same radical pair, same distances, very similar properties, but they show weaker magnetic field effects. Now, that could be due to a variety of reasons, but might be because the robin protein has evolved to be particularly sensitive to weak magnetic fields in a way that the pigeon and chicken proteins haven't, for birds that don't travel huge distances every year. Okay, so that summarizes quite a lot of work, much of which was published in June this year. And we were lucky enough to have that featured on the cover of the journal, where you can read a bit more about it if you want to. So now, let's move on a little bit, just in the last few minutes. A question that people often ask is, if these sensors, if these cryptochromes really are the sensors, and they're in photoreceptor cells in the bird's eyes, does that mean that the birds literally see the Earth's magnetic field? That's a very good question, and like so many in this area, we don't have a very good answer, but it's possible. We don't know what the bird perceives, if this is the right mechanism. It could be some visual impression superimposed on the field of vision. And as the bird moves its head around, some pattern needs to be dead center in that field of vision, and then it knows where the north-south axis is. Well, that might be how it happens. So we don't know about that yet. We would have to devise some clever experiments to do that, but what we can do is to do calculations to imagine what it is the birds might perceive, because we now know quite a bit about the magnetic properties of cryptochromes and how they respond to the Earth's magnetic field. And so we could simulate in the computer what the bird might perceive. And so I'm going to show you a little video which we mocked up a year or two ago, which is admittedly science fiction, but might have some basis in reality, which gives a kind of impression of what might be going on. So what we did was to fly a drone over a cornfield just outside Oxford. And this object was flying around in a circle. And then an undergraduate summer project student who was quite hot on computing, superimposed on this video footage the patterns that we had calculated based on the magnetic properties of the cryptochromes and the flavin and tryptophan radicals inside them. So what you will see is a changing view as the bird scans the horizon, moving around in a circle above the cornfield where we did the filming. There's a compass rose here at top right, which shows you which direction the bird is looking in. And then superimposed on that, you will see the calculated patterns. The colors are red and blue. They don't mean very much. We just colored them to make the pattern look more visible. So let's start the video going. There we are, the bird's looking around. Here's the magnetic patterns starting to appear. The birds are looking to the west now. And as they swing round to the north, a distinctive pattern, here it is, will emerge. Now let me try and stop that when they're facing due north. And there we are, there's this cross-shaped pattern based on what we know about the magnetic properties of the cryptochromes. And so hypothetically, when the birds see this, when it's dead center in their field of vision, maybe they know that's the north-south axis. Well, we don't know whether that's true or not, but it's an interesting, it's an appealing speculation. Sorry, excuse me. Apologies. Okay, so how do we test whether this really is the right mechanism? Well, that's quite difficult. I just want to talk about one possibility. So we know from lots of laboratory studies that the way in which radical pair reactions respond to static magnetic fields can be affected if you superimpose time-dependent magnetic fields. And so it proves also happens with the birds. That is, I showed you those shielded chambers inside the non-magnetic testing facility at the University of Oldenburg. If you disable the magnetic shielding around those chambers such that electromagnetic noise can get in, then the birds become disoriented. It's really quite remarkable. So they experience, without the shielding, they experience electromagnetic noise generated by all kinds of electrical equipment on the campus of the university. So every piece of electric equipment generates very weak electromagnetic noise of a variety of frequencies, including the same frequencies of the singlet triplet interconversion that is crucial right at the center of this radical pair mechanism. So the kind of frequencies emitted by AM radio masts, like this one here on the left. So if we don't shield the birds from these incredibly weak time-dependent fields, sometimes known as electrosmog, we humans are exposed to this all of our lives, unless we're right out in the countryside, away from human activity and electronic equipment. And this seems to disrupt the bird's ability to use their compass. They jump in random directions in the Emblem funnels, even though it's the migratory season and they're exhibiting the migratory restlessness and would normally jump in the appropriate direction for migration. So we don't really understand that effect, but it has been interpreted as a possible diagnostic test for this radical pair mechanism, because it's difficult to imagine that single crystals of magnetite could possibly respond to a magnetic field that is inverting more than 1 million times per second. Magnetite crystals big enough to rotate in the Earth's magnetic field would be far too big to oscillate 1 million times per second. So that could be a way of demonstrating if we could really understand quantitatively how this works. of demonstrating that this might be the right mechanism. Okay, so now to wrap up, my time's almost up. There are lots of open questions. I've mentioned a few of them. I just thought I would list some of them. Not all of them I've mentioned already, just so you don't get the impression that we understand everything. That's far from the case. Lots of things that need to be done. We've been studying purified cryptochromes in the lab. We don't know yet whether they behave in exactly the same way in a photoreceptor cell in the bird's retina. Of course, in the cell, they interact with other proteins, both for signaling, but also they need to be oriented. If you have a molecule that is rotating in solution, then it might be an intensity sensor, but it won't be a magnetic direction sensor. It needs to be tethered, immobilized, and aligned, so the different cryptochromes reinforce one another's magnetic field effects to give you a direction sensor. We need to be able to do some of the spectroscopy in vivo, not just on purified proteins. We don't know how the proteins could be immobilized and aligned in the cells. The photoreceptor cells, which do contain Cry4, seem to be particularly attractive locations because there are, of course, in those cells, membranes which are aligned, in which the opsin proteins are embedded that are responsible for vision. So maybe the cryptochromes are somehow attached to opsins, which are themselves aligned by the membranes in the inner or the outer segments of those photoreceptor cells. That's something we need to investigate. We don't know much about signal transduction. We're only just starting to identify what the binding partners are for migratory bird cryptochrome 4s. The magnetic fields, the magnetic fields that we predict and measure in the laboratory are quite weak. Sorry, I'll call you back. Excuse me. I apologize for that. The magnetic field effects we measure are quite weak. We don't know whether they are large enough that they could form. For God's sake, excuse me. Right, apologies, I should have turned all the phones off before I started. Right, and finally, is there enough light at night? These are nocturnal migrants. Of course, there has to be some light at night. There's the stars. If it were totally black, the birds wouldn't be able to see and they wouldn't fly at all. Is there enough light for this light-dependent magnetoreception mechanism? And how, in quantitative detail, does this radio frequency field effect work? So those are just the few things that we're working on. So birds, if you've ever wondered what birds think about while they're migrating, well, it's not GPS, of course. They don't have sat-nav, satellite navigation, but maybe they have chem-nav. And I think that in the next 5 years or so, we'll have a much better idea about whether this is really the right mechanism of the magnetic compass and whether cryptochrome is the right molecule. So I'd like to finish by thanking the people who did all the work here. I'd like to mention in particular by name my colleagues in the chemistry department at the University of Oxford, Christiana Timmel, Stuart Mackenzie, David Manolopoulos, and Justin Benesh. Also my principal collaborator at the University of Oldenburg, Henrik Mauritsen, a biologist, and also Ilya Solovyov, a computational biophysicist. I'd like to thank them, a large number of very talented graduate students and postdocs in the two universities who've worked on this research for a number of years, and collaborators at other institutions. I thank them. I thank these funding bodies for generous funding of this research, and I thank you for listening. Thank you.
Thank you very much, Professor Peter Hore. And I think I speak for everyone in saying that was truly breathtaking, both the science and the explanation, really. And I thought it was a great intersection of the biology of avian navigation, the physics of magnetism, and the chemistry of radical pair reactions. So thank you for that. So now we're going to have a question and answer session. And as part of that, we will have either questions from the YouTube chat, which I will try and compile for Peter. Otherwise, if you'd like to ask a question on Zoom, please do use the raise hand function, and we will let you speak in turn. Otherwise, you can always put your questions into the chat, and I will ask them myself. So first off, we have Aryaman, who has raised his hand, so I'll just let him ask the question.
So firstly, I would like to thank you for this talk. It was really interesting. And what I want to ask is that in 2019, there was a Nature article about how the magnetic North Pole is shifting towards Siberia from Canada because of movements in the iron in the Earth's core. So does that also affect the way that the birds perceive the magnetic fields? Because surely their migration pattern should remain the same, because they're going from one climate, I guess, to another. So if the Earth's magnetic field is changing, then the migration pattern would also change. And will this have an effect on the way the birds evolve?
Yes, it may do. We don't really know the answer to that. Of course, I hope I haven't given the impression that the magnetic compass is their only source of directional information. I mean, clearly they can use the sun and the stars, landmarks, smell, other cues, clearly. So all of which needs to be integrated in the brain before a decision can be made in which way to fly. So it is possible to recalibrate, for example, the magnetic compass using other information. So the first time the birds fly, it's probably not so crucial to the nearest 10 or 50 kilometers where exactly they stop in North Africa. But it's clear that on the next journey south, they can find the same tree again. But that's because they can then detect the intensity of the Earth's magnetic field and recognize landmarks and so on. So I think that sort of combination of different sources of information would allow them to adjust what weight they put on how they interpret the magnetic information.
Thank you.
So we have a question from Petya in the chat. So Petya says, super interesting talk, thank you. Back when you introduced the reactant intermediate product scheme, there was no way shown for the product to be converted back. Does it accumulate in the receptor cells, and how can this model be reconciled with Cryptochrome?
Yes, so it may need to be reconverted, or it may not, we don't know. I mean, one possibility is that having formed the signaling state and done its job with the signaling, that the protein is then degraded and then fresh cryptochrome is expressed to replace it. That's one possibility. Another is that the flavin radical in the middle of the protein gets oxidized by, gets reduced by some external electron acceptor, and then that returns the protein to its ground state so it can absorb another photon. Now, for the purified proteins, that process is rather slow in the absence of added redox agents. So we don't know how rapid that is inside the cell. So it's a good question, something we need to find out and determine whether there really is a photocycle, or whether the protein gets degraded and then replaced.
Okay, yeah, thank you. We have another question in the Zoom chat. So this is from Makoto. About the mechanism of the axial magnetic compass. Is there any suggestion how gravity affects the magnetic compass?
There's no direct gravitational effect on the photochemistry. That interaction is absolutely tiny, but gravity is important. I mean, the birds need to know to distinguish up from down. Sorry, that may seem rather obvious, but if you think about how the axial compass works, what they're essentially detecting is the angle between the magnetic field vector and the horizontal. So you need to know, for example, in the Northern Hemisphere in the spring, when you need to fly north, you need to detect the direction of the horizontal and the vertical components of the field. So that's with reference to the vertical, which of course is downwards. So, the vestibular system responsible for detecting the gravitational field must be involved somehow in the brain in processing this information, but not directly in the sensing.
Thank you. There is another question in the chat. So I think this is touching on the sort of last point you were making. And it's about, how can they distinguish the magnetic field of the Earth in man-made areas within human developed regions? And so do cities disorient their sense of direction?
Maybe, yes. So the birds I mentioned that were disoriented by the electrosmog on the campus in Oldenburg, if the same birds are taken a couple of kilometers outside the city into the countryside, away from human activity, then with no shielding at all, they can use their magnetic compass perfectly well. So we don't know whether in the wild the birds are affected by man-made electromagnetic fields. So they fly quite high, typically above the cloud level. So it may be that they're high enough that this electromagnetic noise is too weak to affect their magnetic compass. They may also avoid flying over large cities. And there may be other reasons for that, because they don't like the lights or the noise or something like that. So we're trying to do experiments to test just this. So at the moment, it's at the stage of attaching radio transmitter tags, so we can keep track of where the birds fly, releasing them after they have been exposed to different radio frequency field conditions to see if that affects the direction in which they start off and whether it might, for example, delay their departure while they take a longer time to get a good compass bearing. But then it would be nice after that to attach radio frequency tags that not only told you where the birds are, but would generate radio frequency fields strong enough that they might disorient, might interfere with the compass, and then see which way the birds fly. So that's something I think will be done in the next few years.
That sounds very exciting and promising. Thank you. We have a question from YouTube. This is from Matthew. Thank you for the amazing talk. I was wondering if you knew whether the time of year migration happens is influenced by changes in the Earth's magnetic field or changes in the bird's protein production.
Changes in the Earth's magnetic field, I think the answer to that is no. There are small variations in the Earth's magnetic field strength, so on a variety of timescales. So there is a daily variation. It's only a few nanotesla in a total field of 50 microtesla. And that arises from the gravitational effect of the sun on the Earth's atmosphere, which slightly distorts the magnetic field. Some people think that might be the source of entrainment of circadian clocks, but that seems a little unlikely given how weak that variation is. There can be much larger variations in the Earth's magnetic field as a result of magnetic storms. And there's some evidence that racing pigeons, are less likely to return on time if they're raced during a magnetic storm. So I think probably the variations in the Earth's magnetic field, leaving aside the storms, probably don't affect the birds very much. Sorry, I've forgotten the second part of the question.
So it was about birds' protein production over time. And whether
yes. So for the Cryptochrome 4, there is a circa annual variation in levels of expression. So more of that protein is expressed in the spring and the autumn in migratory birds. That's not true of domestic chicken, which don't migrate. And so it is possible, indeed, that there are higher levels of Cryptochrome 4 during the migratory seasons when the birds need it.
And I think another sort of speculative related question here is what happens when the Earth's magnetic field flips entirely?
Okay, well, if it happened instantaneously, the birds would be none the wiser. But that doesn't happen. It takes of the order of 10,000 years fully to invert. We are apparently due for another reversal. There hasn't been one for the last 700,000 years. And so probably sometime in the next 100,000 years, there will be a flip. And as we heard, I think from the first question, there are weak indications that it's beginning with movement in the direction of the field. So we don't really know what might happen to the birds during those periods. But it seems likely that they would just place less reliance on magnetic information. So the integration in the brain then would have a heavier weight placed on other sources of directional information, or maybe there would be some way of, as it were, turning up the amplifier on the magnetic compass sensor so that even though during the reversal, when locally the Earth's magnetic field could be easily 10 times smaller than it normally is, maybe the sensitivity could be turned up during those periods so that the field could still be used in some way.
Yeah, thank you. So we have Tristan, who's got his hand raised, so I'll hand over to Tristan to ask his question.
Thank you very much. Firstly, thanks for the amazing talk. My question would be on the evolution of the Cryptochrome 4. Do you know if this mechanism of magnetoreception evolved just for the sake of magnetoreception, or did it maybe improve their eyesight first and then the birds learned to use it to navigate the magnetic field?
I don't think there's any evidence that cryptochromes are involved in vision. They do have a number of functions. The best understood is in regulation of the circadian rhythm. So in plants, humans, birds, insects, and so on, cryptochromes are one of the large number of proteins that are involved in our 24-hour circadian rhythms. Only in insects do cryptochromes also act as circadian photoreceptors. So human cryptochromes, which don't seem to bind FAD, the flavin, are involved in transcriptional regulation of the 24-hour rhythm. So, and there are other possible functions of cryptochromes. I mean, certainly plant growth regulation, possibly insect behavior as well. So I think that the way it may have evolved is that there are DNA repair enzymes called photolyases, which are the evolutionary precursors of the cryptochromes. They do bind FAD, and they use blue light to repair DNA, which has been damaged by UV light by forming crosslinks. So it's possible that if the photolyases in their DNA repair function accidentally, because they form radical pairs, showed a magnetic field effect, that could have been optimized by evolution for DNA repair, and then that could have been inherited by the cryptochromes, and then possibly exploited by birds, maybe other organisms, for magnetic sensing.
Great, thank you.
That's one possibility.
Thank you. We have another question from the chat from Isabel. So it's, is the arrangement of residues around the chromophore important for magnetoreception to work? For example, if the position of the tryptophan residues are changed, would that affect how the birds orientate themselves?
I think it would change it, yes. So I keep saying we don't understand things, and this is another one of them. The orientation and the positions of those tryptophans are very highly conserved. So not just amongst the migratory bird cryphores, but all avian cryphores. And if you compare, for example, the Arabidopsis cryptochrome with the robin cryphore, then apart from the additional tryptophan at the end of the chain, you can almost superimpose the two. The flavin and the first three tryptophans from almost exactly the same positions and orientations. So it looks like that has evolved to make the electron transfer very efficient. So you've got just the right overlap of electronic wave functions on electron donor and acceptor to get fast and efficient forward electron transfer and inefficient side reactions that would reduce the quantum yield of charge separation. I mean, that's something that we had thought about trying to investigate by, for example, replacing a tryptophan with a tyrosine, maybe somewhere in the middle of the chain. So tyrosine, unlike phenylalanine, does have redox properties to produce radical pairs. Because of its different size, it would have a different orientation, maybe a different position. And so we could perhaps learn a bit more about what controls the efficiency of the electron transfer that way.
Thank you.
Then the differences between the robin, chicken, and pigeon proteins, that must be due to the small number of sequence differences in the residues that surround the tryptophans and the flavins. So yes, that's important too.
Right, thank you. And sort of coming back to the evolution question, do we think that migration sort of evolved earlier than this sort of magnetoreceptory system, or was it the other way around? So did magnetoreception kind of enable migration, or was it just a sort of enhancement of migration?
Yeah, that's a good question. Again, so I mean, as will be clear, I'm a physical scientist. I'm not a biologist, and so I'm not the best person to answer some of these questions. But my guess is that migration came first, and then anything that would increase the probability that the bird would make it all the way to North Africa, say, there would be evolutionary pressure to optimize that. And so I think all sorts of things would have been optimized once the bird started flying large distances. That's my guess.
Yeah, thank you. I think we have one final question from the YouTube chat. But if anyone else has a question, please do put it in now, because we will wrap things up shortly after this question. So there's a question from Aman in the YouTube chat. He says, am I right in understanding that the bird only knows the north-south axis and not the specific direction? And so it would need to integrate other factors like sunlight to know the exact direction.
Yes, that's right. So it needs to know which season it is. So whether to fly north or south. So it gets the axis, but then indeed so there are all sorts of other reasons why the birds need to know that spring or autumn is coming. When they need to prepare their bodies for mating and breeding. And so that means knowing weeks in advance that spring is just around the corner. So for sure, as well as their circadian clock, they have a circannual clock. So the sensors for that are opsin proteins deep in the brain. And so the birds have a separate system for detecting changes in day length. That will prepare them for the physiological changes they need for breeding, and also alert them that the way they interpret the compass information is that they fly north in the spring and south in the autumn.
Thank you very much. I think that wraps up our Q&A session as well. So I'd just like to thank everyone who came to this event. So thank you for turning up on YouTube. Thank you for coming in on Zoom. And just before you all leave, I'd just like to remind you that the student speakers for this session will be starting at 11, not 10:45. So please do come back in about half an hour, 45 minutes to see the student speakers. And of course, a huge thank you to Peter. And we'd love to host you in Cambridge sometime soon. So thank you very much.
It'd be nice. Thank you very much. I enjoyed it very much. Excellent questions. Thank you.
Thank you. We will just stop the live stream.